Blade Runner. Ridley Scott's epic masterpiece imagines a dystopian future where rebellious artificial intelligence systems create the need for a specialist police force to exterminate them. From our perspective, Blade Runner is set in the past, in 2019, in a city that resembles modern-day Kuala Lumpur. The fusion of artificial intelligence, robotics and biochemistry never quite happened. However, Blade Runner offers a possible future, or perhaps an alternative present. The film explores the nature of sentience and slavery. Scott's film noir presentation, combined with Rutger Hauer's poetic and brilliantly paced delivery, produce a film that exceeds the boundaries of mere science fiction, making Blade Runner one of the greatest films of the 20th century. That is, until we take a sharp look at the underlying science. Aware that the replicants are liable to turn nasty once they acquire memories and unusual emotional responses, the Tyrell Corporation restricts their lifespan to 4 years. In the scene where Roy confronts Tyrell about overcoming the lifespan issue, the actors courageously battle through a script that is in fact utter nonsense. It rambles on about mutagens and viruses and gets thoroughly confused. Although the script was written 45 years ago, the dialogue was nonsense even then. It's always easy to criticise, but one of the challenges in scientific script editing is finding a way of making things work and design a script that the actors can memorise and deliver effectively. How might the dialogue be written today given the advances in genetics and biochemistry in the last half-century? In other words, how might we build a replicant? The easiest method of doing this on an industrial scale is cloning. However, until recently this was a formidable challenge. We thought we understood genetics, yet cloning techniques repeatedly failed. It turns out there was another level of gene activation called epigenetics. This is also inherited. Most gene expression can be finely tuned by alterations to the histones, a set of proteins embedded in the chromosomes. Adding a methyl group to the key sequence stops the gene altogether. These markers can be altered or removed during the lifetime of an organism. This is a potential problem for a mass-market genetically engineered product. The pattern of genes coded in DNA isn't really like a blueprint for creating an organism, it's more like a film script. A Star is Born was filmed 4 different times over 80 years. Although the plot and the characters are the same, each version is quite different according to the screenwriter's handling, the director's vision and the performance of the actors. Similarly, the expression of genes via epigenetics can lead to quite different characteristics, even though the genetic makeup is the same. Since epigenetic expression is a tad unpredictable, a 4-year lifespan might not seem too bad an idea. The team that created Dolly the sheep, the most famous mammalian clone, had immense difficulty in getting the process to work at all. 300 times they painstakingly replaced the nucleus in a sheep egg cell. All but one attempt failed, and even then Dolly had premature arthritis. A few years later, however, researchers developed a process to create induced pluripotent stem cells, or iPS cells. This was the key breakthrough that allowed any cell to be converted into almost any other type of cell, depending on how the epigenetic gene expression is handled. At last, there was a theoretical method to build replicants. Their ability to handle extreme cold and heat is an interesting feature. Perhaps genes from extremophile bacteria might allow this. These bacteria have evolved some useful enzymes and proteins that enable them to survive in conditions that would rapidly kill normal cells. It is thought that the early archaea developed this type of metabolism early in Earth's history, and subsequent multicellular organisms lost these abilities as Earth's environment became more stable. Inclusion of these genes to make these proteins in the replicants would be a handy feature and most likely to be compatible with existing cellular biochemistry. But how can Tyrell restrict the replicants to a 4-year lifespan? Tyrell's concern that the replicants' increasingly autistic-like behaviour, developing unusual emotional responses and gathering appropriated memories would make them difficult to control as slaves. Assuming that the replicants could be grown very quickly in some sort of mad cascade of cell division, then some means of halting cell division close to the desired 4-year lifespan would be needed. Normally the number of cell divisions is determined by telomeres. These are short repeated sections of DNA at the end of the chromosomes. These serve as a stop signal to the enzymes controlling DNA replication. Each cell division removes a section of telomere. If the telomeres are engineered to an ideal length and are rapidly dividing tissue, such as skin, lungs, or gut, these tissues would collapse when the limit of divisions is reached. This might explain the white encroaching discoloration on Roy's face and hands. The dermis can replicate no more and the skin is replaced by scar tissue. Theoretically, a sudden cessation of cell function would resemble radiation sickness. The skin, gut and lungs would just slough off and the result would be rather messy. However, in the film, Roy just dies due to some sort of internal neurological crisis, which seems to affect his hands first. Since nerve cells tend to live quite long with slow division, telemetric shortening won't work. However, if some of the more rapidly dividing neurological support cells, such as glial cells, are restricted in this way, then it would be possible to disrupt and destroy brain and nerve function. With this in mind, we can now rewrite the dialogue. Tyrell could explain that lengthening the telomeres in each of the billions of skin, gut and glial cells would be impossible. Roy counters with the suggestion of a messenger RNA vector that can code for telomerase, an enzyme that can grow extra units on the telomeres. Tyrell objects by saying that the random hodgepodge nature of telomere addition would cause malignant cancers due to the rapid cell division genes. Roy comes up with the idea of an epigenetic block to the relevant genes, but the difficulties of replacing billions of components prove to be too difficult. The outcome is the same. Roy kills Tyrell in a fit of rage. Some of the other science fiction concepts in the film can be safely ignored. To see Roy's attack ships on fire off the shoulder of Orion would require a round trip of 245 light-years to Bellatrix. No mean feat for Roy in less than 4 years. Perhaps the off-world colonies are much closer to home. The solar system offers a range of environments that would suit genetically engineered beings, particularly ones adapted to extreme cold. In many ways, too much attention to scientific detail detracts from the overall thrust of the film. It might have been better if the writers had left these details rather vague in the original film. However, a small tweak can establish scientific accuracy without disrupting the overall brilliance of the film.